Hi everyone. So today we are going to learn about Arabidopsis thaliana. So in any of the plant molecular biology lab, we spend a massive amount of our time focusing on invisible stuff such as cloning vector, analyzing RNA-seq data, or troubleshooting CRISPR-Cas9 wheels. But today I want to talk about the physical foundation that makes all of this work possible, which is the cultivation of Arabidopsis thaliana. Why do you think that learning this is important? Because a single stressed plant can introduce enough noise to ruin weeks of your delicate lab work. So we will walk through the life cycle of this model organism from a dormant seed to a successful harvest and ensuring that all the plants are as robust as the data that you're trying to collect. So let's dive into this. So you all must be thinking that why we consider Arabidopsis thaliana only as the model plant. Why not any other plant, and why only this plant? So the answer lies is that this plant is very efficient and predictable. So since 2000, we know the genetic map of this particular plant, which is around 135 megabase pair genome. But the real value lies in its life cycle. So there are 4 or 5 points that are very important. Then why do we choose this plant? First is its small size. It has a rosette diameter of less than 10 centimeter and plant height of nearly 30 centimeter. Secondly, it has a very short life cycle. For example, it can grow from seed to a complete harvest within 6 to 8 weeks. Then it is very easy to grow. It doesn't have very sophisticated growth requirements, and it can produce up to 10,000 seeds per plant, which is quite a big number. So that is why people or the researchers consider Arabidopsis thaliana for studying such variations or any of the plant molecular biology stuff. So before we touch a single seed, we need to talk about the substrate. For all developmental studies, we rely on Murashi and Skoog's media, a precise cocktail of macro and micronutrients. We solidify this with a high purity agar, which can be phytogel or agarose, to give us a clear view of the root architecture. But here's where it gets practical. For soil grown plants, we generally use a mix of sphagnum, peat moss, and vermiculite. This ensures proper aeration, moisture retention, and why do you think this matters? It is because consistency in the growth of media is the first step towards reproducibility. If your soil varies, your phenotypic data will also vary. So it is important. So before we begin with our experiment, it is very important that we store the Arabidopsis seeds in a proper environment. So we need to make sure that the Arabidopsis seeds are labeled properly and the year should be mentioned over there. Another important thing is that we should store them in a closed environment. We should label them properly, and it is very important that we store them in a moisture free environment. Why? Because it is important that moisture should not be there, as it may trigger metabolic leakage where the embryo slowly burns through its stored lipids and protein. So what we can do is we can take Arabidopsis seeds, keep a desiccant, and store it at 4 degrees Celsius. This will keep the seeds safe, and it will help in maintaining a homogeneity when we start an experiment. So as we now have the seeds, we need to start the experiment. For that, we need to sterilize the seeds because the seed outer surface may have different types of organisms, and we are growing the seeds in the MS media, which is a hub of nutrients. So microbes might grow very well on the MS media. So to avoid that, we need to get rid of the external microbes that are present on the seed surface. For that, we do surface seed sterilization using chemical sterilants. So first we take some Arabidopsis seeds in an Eppendorf. The first process is to wash the seeds with ethanol. Now the duration may vary according to your experiment. We don't want the seeds to get fully dry because this takes out the water. So first we rinse at a particular duration with 70% ethanol. Then we wash the seeds again with water. Now the next step is to clean the seeds with 10% bleach for around 5 to 7 minutes. You can just gently tilt the epindoff up and down for that, and then we wash the seeds with sterile distilled water for around 3 to 5 washes. Once that is done, we remove all the water. We are left with the fresh seeds that we can use for plating on the MS agar media, and all this is done on a laminar flow cabinet, which provides us the aseptic conditions. So once the seeds are plated, we do not go straight to the growth chamber. Instead, we return them to the dark at 4 degrees Celsius for 2 to 4 days. This process is known as cold stratification. But here's a surprising part. We are essentially tricking the plant's internal clock by mimicking the end of winter. We trigger specific hormonal shifts like decreasing of abscisic acid, which inhibits growth, and increasing gibberellic acid, which promotes it. Why does this matter? Because it ensures that every single seed germinates at exact same moment. In research, the synchronized germination is the secret to reducing statistical noise and keeping your error bars small. Yeah, so once we are done with storing the seeds at dark and cold stratifying them, we move the plates into the light. We usually maintain a temperature of around 22 degrees Celsius and light intensity of 120 to 150 micromole per meter square per second. Now, this is where it gets interesting. Within the first 24 hours, the radicle emerges, making the transition to active growth. By day 4 or 5, the cotyledons unfurl and begin photosynthesis. Why does this matter? Because this is the most fragile stage of the life cycle. If your light intensity is too low, the seedlings will undergo etiolation, stretching their hypocotyles in desperate search of light. We want compact, sturdy seedlings. For that reason, the light intensity matters a lot. So this is just an overview of how Arabidopsis seed germination occurs. So we have the Arabidopsis seed on MS agar, which is the day 0, no radical emergence, no plumule there. So we have now the radical emergence within the first 24 to 48 hours. Then the cotyledon growths happen within 3 to 4 days, and we have healthy Arabidopsis seedlings, young seedlings at 5 days. We need to make sure that the temperature is controlled around 22 to 23 degrees Celsius, and the light intensity is about 100 to 150 micromole per meter square per second. So once we have the seeds germinated, now the next step is to transfer the Arabidopsis seedlings to soil. So here in the MS agar plate, we have the Arabidopsis seedlings. We need to carefully lift the seedlings, and like we need to avoid root damage. Okay, so carefully we need to pick the seedlings, and as we mentioned earlier, we need a potting mix. So we use the little wet or moist potting mix and transfer these seedlings into the soil without damaging the roots of the plant, because roots of the seedlings, because if once they are damaged, the plant might not survive. So here I have just given you the steps of how do we transfer a Arabidopsis seedling from MS agar plate to soil. So we carefully pick using the forceps. We have the forceps, we pick up a healthy seedling, and then we kind of transfer that into a soil mixture. We press it gently insertion and soil closure happens, and we just sprinkle some water over the plant. So we usually, in a plant molecular biology lab, we usually keep our plants in a plant growth chamber. So why growth chamber is important? Because it provides controlled light cycle, it provides controlled temperature, and it also controls humidity. So we can keep our Arabidopsis seedlings in the shells, and we can neatly label them so that we know what is the progression of the life cycle of Arabidopsis for our experiments. So it is very crucial for us to maintain the plants in the lab, and there are few factors that might affect the plant growth. So here I'm going to mention things that we need to avoid for proper root care of Arabidopsis. So we need to make sure that the water that we are giving to the plants is well drained. There is no clogging of the water within the pot, because that might make the roots of Arabidopsis rot within. Make sure that there is no water logging, you are not overwatering, and there is no fungal contamination. Once, if you observe that there is any fungal contamination, you should quickly remove that plant so as to avoid that fungus infecting other healthy plants. So once a plant finishes the vegetative stage wherein it grows its rosettes, it enters into bolting, which is the reproductive or flowering stage of the plant. So here we know that we have the rosette leaves. From this, a single stem arises, which we call as the bolting stem. Now it may have many branches of the stem arising from it. At the end of each of the stem, we may have some flower buds. Then we can also have flower seed pods, which are known as silk. Okay, and this is how do we know that a plant has entered into the reproductive stage? Is when we see the bolting stem, flowers, silk, and flower buds. Okay, now Arabidopsis silk formation and seed collection. We are going to study about this. So we need to harvest the seeds, and if you are doing any of the genetic experiment, we need to keep the seeds of different lines separately. So here we have the Arabidopsis inflorescence, and here we have green silk. Here is a mature Arabidopsis plant. Where once the silk starts drying, we have the mature silks. We can collect these dry silks and brush off the seeds and store them into different packets with proper labels for our experiments. So here we have some golden rules of Arabidopsis lab. Even with the best equipment, some lapses in technique can lead to big errors in your data. So here are some checkpoints that we need to keep in mind when we are growing Arabidopsis. First thing is that we need to follow aseptic technique to avoid any of the fungal contamination. Next is we need to properly label all our samples so that we are not mixing them. Then next is over sterilization. We need to be very precise with the timing of the chemical sterilants that we use to treat our seeds with, because over use of the chemical sterilants or increasing the duration might affect the seed germination. Coming to the fourth point, we need to avoid overcrowding of the seeds because that may lead to stunted growth of the plants. Next, we need to avoid overwatering because that might cause root rot. And finally, we need to maintain clean conditions for our plants to grow so as to avoid any contamination by mold or bacteria. So if you stick with these precautions, and you can set your experiment to success. So now wrapping up our study or the presentation, here are some 10 critical steps that we need to follow. First is seed storage. We need to ensure that we store our seeds at 4 degrees Celsius in properly dry conditions in a cryo box. So for the experiment, we need to make sure that we are sterilizing the seeds with mild chemical sterilants, and we need to keep in mind the duration of the sterilization process. Then we carefully sow the seeds on the MS agar plates. We cold stratify the seeds so that there is uniform germination. Then the germinated seedlings we keep in proper light conditions um, and proper temperature for their growth, uniform growth. Once that is done, we transfer the seedlings into soil. We take good care of the growth in the pot, like we should avoid overwatering the plants, and we should avoid the root rot. There should be proper drainage system for the plant. Next, we need to observe the plant still flowering, and then we need to carefully look at the silk formation, and finally we need to see the seed collection. And that brings us to the end of our guide. Growing Arabidopsis is as much an art as it is a science, but with these protocols in your toolkit, you are ready to produce high quality reproducible plants for your research. If this walkthrough helped clarify your lab work, please give this video a like and share it with your fellow researchers. Don't forget to subscribe to Rooted Science for more deep dives in plant molecular biology and lab techniques. Thanks for watching and good luck at the bench. I'll see you in the next lesson.